So let me start this again. This is Engineering 443, 543, Computational Modeling. This is lecture 1. I need to remind you that this class is live streamed and recorded, so please keep that in mind. You'll have access to it. Through YouTube if you want to watch the lectures after the fact. And again, we will be probably offering this primarily in the mixed format for people who can't attend in person or who choose not to, if they don't want to park, for example, or they are on the other side of campus. And we'll see how things go during the semester. There may be possibly 1 or 2 weeks when travel means that it has to be virtual, but we'll see. To begin with, I have some handouts. I don't know if people do handouts anymore, so I did not print out that many copies of this because I figured not everybody would want a paper copy. But in case you do, everything is available online. I have here some Python documentation on CompuCell. This is a set of beginner's Python introduction. And you'll have to step through it and find out where they break. This is a bit of an introduction to modeling in general. And print out a homework number 1. And so if there is a need for extra copies of this, I'm happy to print them out, but I didn't want to print out 16 copies of 100 pages and then have nobody take. So if you're one of those people who prefers to have things online, everything that is being handed out as printout has been given to you electronically. But if you feel more comfortable with a paper copy, you've got it. So today we're going to be talking about, and this semester we're going to be talking about modeling living organisms. And we're going to be modeling living organisms from the point of view of cells and how they work and how they organize. And a few people in the class, I think only 2 or 3, were in the class last semester, which covered what happens inside of cells, modeling what goes on inside of cells. And this is, at least conceptually, a bit of a sequel to that, although people very often take the courses out of order. There's no prerequisite that you have to take these 2 courses in order. But we're going to talk about how organisms organize themselves, how they maintain themselves, and how they fail, and how you model that. And so today I'm going to go over a little bit of logistics about how the course works. And then we are going to start jumping into doing a little bit of simulation. And then we will come back and do a little bit more perspective. And it's critical that everyone have, as I asked online, everybody needs to have a laptop here. You're going to have you need to have your laptops available because we're going to be doing coding during class. So if you don't have one this week, then lean over your neighbor, I guess. But going forward, it's important to have it. This is going to be a project-based class. You're going to hear that a lot. The people who've taken classes with me before know about that. 16 weeks is not a lot of time to do a project. And so you're going to get pushed to think about projects from the very beginning. And I realize that that's not always an easy thing to do when you haven't done the material before. I'm going to be available. Hayden, who's online, will be available. Giuliano will be available to talk to you individually in groups, trying to think about projects. And I hope that we can come up with things that are interesting and fun for everybody. since it's a big class, that's good. It means we can have slightly larger teams, and that reduces the workload on individuals. And it also means that if we have people with different backgrounds, sometimes we have people who are more biological, sometimes we have people who are more mathematical, sometimes we have people who are computer engineers. It's nice to try to build a team that combines different perspectives and skill sets. So we're going to be meeting. There was some ambiguity in the registrar's office, actually, about exactly when this class meets. I think officially we're meeting 4:45 to 7:45. The schedule earlier in the registrar said 4:55 to 7:55. I can't ask if I ask you if you were able to get on by Zoom, that doesn't really do any good if you weren't, since if you're not here, there's no way I can talk to you. We do have an associate instructor for the class, Giuliano Ferrari Gianlupi, who is going to be an expert in this kind of thing. He's just finishing his PhD this coming semester. He also helped teach last semester for people who are in the class then. We'll have Hayden Fidel helping us out, and Joel, who's another graduate student, will also be helping us out informally. For the CompuCell software itself, our resources are Dr. Maciek Swat and Dr. TJ Sigo, who are always going to be available to help us if there are issues with the software. Maciek is in Colorado. TJ was here as a postdoc and just started a faculty position at the University of Florida doing this kind of modeling. And you're going to have online student materials folder. You'll find I'll try to use Canvas, but I really find Canvas rather rickety. And so I prefer to use Google Drive. And so there will be a Google Drive. I hope everybody has access to it. Trying to put up QR codes is a little bit new for me. I'm not used to creating them, but I hope they're helpful so that people don't have to type long URLs all the time. So there is a student materials folder, which everyone should have access to. It will have a variety of things, certainly the things you need, possibly some older material that may not be exactly what you need. Everybody should check to make sure they can get to it. If you can't, let me know by email or otherwise. There are also YouTube recordings of last year's lectures. The first couple of lectures will probably be very similar as the semester develops. I try to adjust the course to respond to people's interests and backgrounds, and therefore the lectures will probably diverge from the older ones as we go forward in the class. But if you want to, there's a playlist last year which you have access to as well. Any questions about that so far? No. Okay, course information. This is a project-based course. There'll be more or less weekly homework, slightly less than weekly, probably. Certainly at the time that we get to the end of the class, you'll be working on projects. I don't want you to be too distracted from your projects by doing too much homework. Last semester, the student said, you know, that last homework assignment is too much for us to do on top of the project. And my response was, we're canceling the homework assignment. So the purpose of the homework assignment is primarily to help you learn. You'll find that they are reasonably open-ended. That means that they can take a lot of time. I don't expect you to spend your whole life on this course. It is a time-consuming course, but it is not meant to be the only thing that you're doing. Therefore, I do expect people to make an effort on homework and try. But if you can't get it done, you don't have to get a perfect score at every homework to do fine. That's understood. The project simulations are going to be released on nanoHUB. People who've done this before know that that's a non-trivial thing to do. The good news about a nanoHUB release is that it counts as a publication on your CV. It's something you can point to employers or to further study. We've had people over the years who've put it on their CV and gotten jobs at places like Google or Facebook based on their class project. So it's worth doing a good job, but it is work. That's another reason to work on projects in teams, because that process of getting things up on nanoHUB takes some effort. I'll be asking people to do oral presentations of their projects and intermediate results. I think you should get credit for that. With this larger class, it may not be so easy to call on everybody every day, but I do expect people to be present, whether virtually or in person, and participate. Helping each other out has been a big part of this class over the years. As I say, people come with very different backgrounds. It's important if somebody's stuck that we all work and help people make progress together. One thing that we did last semester, I think quite successfully, was I'm asking you towards the end of the class, there will be 2 rounds of presentation. You'll do a presentation of your projects. You'll have critique sessions with your classmates, and then you'll have an opportunity to revise the projects and do them again. That's a traditional thing to do in, say, an architecture class, and many kinds of engineering. Computer science, maybe not so much, but if you've had that experience before, you'll be familiar with it. If not, I hope it's helpful. Usually, if people take that seriously, you wind up with a much better final project. We used to say that you had to do a video introduction to your project. In fact, that's asking a lot, so it's going to be an extra credit option if people want to do it. Some people have not had a lot of Python before. We don't do a lot of Python programming in this class. We do a little bit. And if somebody needs to do background Python, I like Rosalind, the online CompuCell, the online Python course called Rosalind, which is a free course. There are a bunch of other online things that you could do. If you have not done any Python and you want to complete a little certificate Python course at the beginning of the class, I'll definitely give extra credit for that because I know it's work for people to do it. In those videos, we have in our summer version of this class, we do a 1-day Python boot camp with about 6 hours of lectures on basic Python and exercises. Those are on YouTube. If people want access to those, they're available to you anyway, but if people have trouble finding them, let me know and I'll point you to them. So, and again, I hope people will help each other there. So class participation really is important. Said that already. Some things that have come up in past semesters, I don't expect there to be any kinds of problems, but I do want to outline this at the beginning so there are no doubts about this. It's fine for people to work together on homeworks. The homeworks could be time consuming and they could be sometimes a bit confusing for people. But I really would ask you not to just hand in a Xerox of somebody else's homework. Especially, I would not ask you, if two people sit down and work together and you're both learning, that's great. If one person does the work and somebody else just copies it, I don't think anybody will do that. But if it happens, you haven't learned anything and they're carrying you in a way that's probably not good for them either. And so I would ask you if you work with somebody, say so on your homework assignment when you hand it in. And also, at least for the discussion of your results, do it on your own. So if you want to sit down with somebody and bang out the coding together, that's fine. But when you're talking about understanding it, please try to do that on your own. I understand that there will be some fluctuations on that, but I think that's important. On the projects, because we're going to have teams, it's fine if somebody says, gee, I'm an experimental biologist. I've never done coding. I really understand how to read bio papers. Another person says, I've done lots of Python programming, but I don't know any biology. If you work together, that's great. but do indicate who did what and make sure that everybody understands it well enough that you can answer basic questions about everything. It doesn't mean you all have to be experts in everything, but do try to make sure that there's some balance in a project. And I'm sure most of you have done team projects before, and so you're familiar with this, but I'd rather address these issues today than have it come up at the end of the semester. Definitely, I'm not somebody who likes a lot of rules. I really like things to be flexible and to make this a joint learning experience. I should say again, when we do this in the summer, it's a free course. It's not graded. There's no course credit for it. It lasts 8 days, 8 consecutive days, 8 hours a day for 8 consecutive days. We went virtual, of course, in 2020 by default. We used to do it in person here in Bloomington. And going virtual had some tremendous benefits. Some losses too, because it's not as you gain. But some tremendous benefits. We were able to get people from India, from Australia, from Africa for the first time, from Europe. We had people basically from South America, all participating in the class together, which was pretty neat. We had 60 people show up. And we did a hackathon for the last 2 days of the class. And much to my amazement, at the end of 2 days, people had some incredibly good projects. After 5 days of 1 day of Python boot camp, 5 days of CompuCell, and 2 days of hackathon, people really actually got a lot done. So I think that it's a different format from here. Here we have 16 weeks. We could go into more detail. We have a little bit more time to breathe. But those lectures are also available online if people are interested. And I think the democratization of modern programming, I think, is very important. Making these things available is important. And that diversity is also important. We had teams that had, in our hackathon, that had somebody from India, somebody from Australia, and somebody from the Netherlands working together. Of course, they didn't get any sleep, but so today is a very introductory course. Then we'll start working on the basics of how you build cells and make them do things. And as early as possible, I do want to start talking about projects. Something that is in the project assignment, I would ask people to look at that project assignment, which I sent out in the email. Please do it as early as possible. And I know it's long, and there's a temptation to just say there is a project and skip the details, but please do read them. And it's fine. I know a lot of people may not have worked in this kind of modeling before. And if a group of you come together and say, you don't know what you want to do, that's fine. We have some ideas to suggest. We can meet with you next week and go over those. I also created a Google Doc, which I think everybody should have access to, where you can put your name, contact information, and a project idea and try to build a team. I don't know how people have done team building otherwise, but we did that last summer for the hackathon. It worked quite well. And so I encourage you to do that. There are lots and lots of, there's no shortage of possibilities for the project. So we'll start out by how to make cells grow, divide, move. Then we'll talk about chemical fields, how to make them work, how to have them interact with cells. Giuliano will talk about how to put things up on nanoHUB, how to do that. That'll be on the 21st, because that's a day I know I'm out of town. And then I've got some weeks listed as additional topics, because that will depend on people's interests. And then preliminary projects, since it's a big class, we may have to do those in 2 weeks. So basically, the hope is that by the beginning of April, you have something that you can present as a preliminary result. And then, of course, the end of the semester is the end of April. Okay, any questions about course logistics before we move on? Was that more or less what people were expecting, or is there something that I don't mean to do, I don't want to spring surprises on you. I may spring surprises on myself sometimes, but I'll try not to surprise. Question, yes. So I sent out in an email and also shared a link. I put it in a Google Doc. So there's a link to a Google Doc that I sent out. And if you can't find it, let me know and I will dig it out for you again. Hayden, could you find the Google Doc link and send it to the class possibly?
Yeah, sure.
So the reason I put it in the Google Doc was that there's the project assignment followed by, here, let me, I have it open here.
Was it a specific link or just the student materials folder?
No, it's actually the upside down. It's actually the assignment. Okay, can everybody see that all right? So what this should have been shared with everybody. It should be available to you. It is, I put it in a Google Doc because people are continually suggesting additional possible projects. And so there's a list of possible projects at the end, and I wanted it to be a live document where when I update it, you have access to it. And if I put it in a Word doc, then it's not, you can't see it live. I wanted it to be available. Essentially, the way this project will work, this was a little bit confused last semester, so I want to be clear about it, is there will be a write-up. It could be sort of a term paper-like write-up, a journal paper style write-up. Somebody said that saying journal paper rather than term paper sounded intimidating. I actually think it's the opposite. Journal papers tend to be shorter than term papers. But anyway, it wasn't meant to be intimidating, explaining what you're trying to do. In particular, what your learning objectives are, because these are educational projects, to some extent, what the biological mathematical concepts are, expected results in further reading. Then you need to actually have a version of that that can be put up online so that somebody who uses your code can understand it, which may or may not be the identical thing, depending on how you want to do it. The code itself and a set of exercises, because when we use nanoHUB, you'll find nanoHUB is a resource for distributing software, but you have to then design things that people can do with it. If you put software up and there's nothing to do with it, nobody will use it. So that's part of the exercise. And so those are the main assignments. Then you'll do a presentation. You have to do a little bit of an advertisement page, a splash page for your application. And then if you want a video tutorial, those are really nice, but I know their work. And I say that I'm asking people to try to think about a project by the end of week 2. That's not because I assume everybody will have decided by the end of week 2. But in my experience, if we're not talking about it by week 2, you're in week 8 by the time you've decided on your project, and then there isn't enough time. 16 weeks goes by awfully fast. And again, I encourage you to team. 3 people is ideal, probably. I know some people like working on their own. I'm not going to prevent you from working on your own if you want to. You've got 4 people, that's okay too. But we have a mixture of graduate and undergraduate students. It's good to have a team that crosses that line. We have some people who have different kinds of backgrounds, more biological, more computation. It's good to cross that background. Well, if you have friends you really, really want to team with, that's okay. But I would encourage you to reach out, maybe work with people who you haven't met before. Just the way in that summer course for the hackathon, we had people, from different continents who'd never heard of each other working together. Learning from each other can be really helpful. The most difficult thing, I think, is always to decide what kinds of things you want to work on, whether that's something to do with embryonic development, cancer. I know there was somebody who said that they were interested in bacterial signaling and quorum sensing and type switching in bacteria. That's a great problem. People have done a lot of modeling of that kind. So those are all possible. One thing that I will say is that people often, I think, try to be a little bit ambitious, maybe too ambitious. And again, I don't want to discourage ambition. If you see this as something you might use in a PhD thesis, that's fine. You're welcome to do that. But actually creating a model that can be distributed, documenting it, creating exercises and putting it online takes quite a bit of effort. And so the model itself is not by any means the only thing that you're doing. And so I would encourage you not to make your life harder than it needs to be, maybe the way to say it. That's all right. So one possibility is to take an existing CompuCell model that's of interest and then create exercises around it and put it up on nanoHUB and distribute it. That's probably in some ways the least ambitious thing to do, but it's fine. Another would be to use a model that uses the base computational methodology of CompuCell, but not CompuCell. There's a great package, for example, by the University of Dresden, Technical University of Dresden, called Morpheus, which a lot of people use. There's some beautiful models written in Morpheus. You could replicate them in CompuCell. I actually mentored a student at University of British Columbia last semester who was doing that for a class project in Vancouver. I know that there are people here who are interested in PhysiCell, Paul Macklin's methodology. That has some very fundamental, interesting differences from CompuCells. And so developing parallel projects in CompuCell and PhysiCell is quite interesting. You could take existing PhysiCell models and implement in CompuCell. You could take a new project and implement it in both if you're in both classes. Those would be reasonable. Giuliano, a graduate student who's a TA for the class, is interested in that particularly, so I'm sure he'd be happy to help people with that. If people are more interested in this kind of modeling for research, it's fine to pick a new biological system and build a model of it from scratch. I certainly don't discourage that. It takes a little bit more effort and background, but if you have a particular thing that you're really interested in that you want to do, that's fine. And then the last option would be occasionally we get people who come into this class with very, very strong computer engineering backgrounds who really want to develop core code. That is hardware and the software extensions of CompuCell as a package. And so I won't say I strongly encourage you to do that. But if you see yourself primarily as interested in doing code extension, we certainly are not. It's an open source software package. We are delighted to have people extend it if people want to. I will say that tends to be a lot to do in a semester, but we've had people try to write GPU accelerators for CompuCell, MPI versions of things and so on. So if people have a real interest in doing that, what you find when you do that is that it's a little bit orthogonal to the class, because in class we're going to be using CompuCell. We're not going to be writing core code. And so it's a little bit more work to do it, but we've had people do very successful things. Thinking of somebody who's now at Purdue in their PhD program who did that one semester. We've had people who've done that. And that code often, although not always, winds up actually being part of the core release once it's been debugged. So are there any questions about the base assignment here? Obviously, there's lots of text here to read, but is the basic outline of the thing reasonably clear to people? Anybody? I'm looking at the chat. Anybody have any questions about that? No? Gabriel,
Yeah.
sorry.
Yeah, sorry. James, it's really good to see you. Happy to be here. But I was talking with Joel, and I was wondering would it be okay to work with Joel? Because we've been talking about, I mean, obviously a lot of our research interests overlap. But I was just wondering, I don't know how if I could work with somebody outside the class on a project, especially. Just wanted to check in.
Sure. Yeah, I mean, that would be fine. You'd be welcome to do that. I mean, there might be, depending on the project, there might be other people who are in the class who'd like to work on that as well. I think what I'd ask you to do is to put that up in that discussion document as a possibility. Put your name, Joel's name, and the topic down, and see if that appealed to anybody else. Don't close off your classmates, but that would be
Oh,
fine.
yeah. Yeah, absolutely.
Great, good point. Anybody else? Peter. You beat, is there a wish? Is there a punch list for what needs to be added to CompuCell? Oh boy. The GitHub repository has a list of pull requests that is so long it's almost embarrassing. Everything from, we'll be using a tool called Wizard that helps you define model specifications quickly. That could be extended. We've been trying to write parallel diffusion solvers. That could be extended. There are energy functional terms that define the core internal computation of CompuCell, where, for example, the Morpheus package from Dresden does it a little bit differently from the way we do it. It would be great if we had the same thing in both languages. PhysiCell has some nice features about describing cell cycle, which Giuliano's been working on, trying to implement for CompuCell. He definitely would love to have somebody work on that with him. So if you want to sit down and go over possible extension packages, that would be great. Soumya, who took this class last year, is a master's student, has been doing extension work for CompuCell. It was a little bit easier to do that when TJ was here, just because, although actually TJ lived in Indianapolis, so for most of his time, so he wasn't actually physically local here anyway, so it was virtual. But the fact that he's moved to Florida doesn't mean that he's not working on the project anymore. So it's definitely possible, and Maciek is available. So if you wanted, if there were a team or individuals, a team that really wanted to do core extensions, I'd say the main thing is to get together, set up a meeting with me, TJ, and Maciek as early as possible to go over a list of possible extensions. I'm not sure that we'd have to look at the GitHub repository to see whether the list of pull requests is public or not. I suspect it is, but I don't know where to find it. So I tend to put things on the stack, but I actually don't know how to look at that stack. Great. Any other questions? Okay. How many people are coming into this class with an idea of what they might possibly want to do with the project? And it's fine to not have an idea. One thing that I want to say about both class participation and other things in this class which depending on your background may or may not be comfortable to you, which is if you're asked a question or you don't know something, it is fine to say, I don't know. It's really okay. If I say, so-and-so, could you tell me this? And your answer is, I don't know, that's fine. And one of the things that we've done over the years, it's a little bit harder in a split class like this, but it works pretty well when you're all in the room or all online. It works okay online in mixed class too. Is when you're coding and somebody gets stuck, it's okay to say, hey, classmates, can you help me out? For a class that's this big, we probably would create breakout rooms and put people in groups of 4 or 5 to work together to debug things. Sometimes we can put things up on the board and work on them together. It's okay to not know. And you'll hear me say, I don't know, and answer the questions. Again, probably a lot of people are comfortable that way anyway, but sometimes it's not easy to say, I don't know in front of a class. And nobody should feel embarrassed for not knowing something. Really, it's okay. Right? All right. So come back. All right. One of the problems for this class is that there is, in fact, no textbook on this subject. Paul or I or somebody else should write one, but we get busy and we haven't done it. We've been promising to do it for years. The closest thing there is to a textbook on the methodologies we're using is about 10 years older. It's Scianna and Preziosi, Cellular Possible Multiscale Extensions of Biological Applications. Some of which use CompuCell. You'll actually recognize on the cover some pictures of an old version of CompuCell. But there isn't a formal textbook. Depending on what you're trying to learn, I really like Gilbert Developmental Biology, biological background. If you want to learn biology and you're not coming from a biological background, I have a very simple secret for you. First, Wikipedia often has good articles, good place to start. Secondly, I strongly recommend buying 10 or 15-year-old versions of the textbooks. You can get a 15-year-old version of Gilbert's Developmental Biology for $5 on Amazon. Developmental biology hasn't changed in 15 years. What we do now is we know a lot more molecular species. And so the textbooks in biology have gone from this thick to this thick to this thick to this thick to this thick. And there's more and more and more detail in them. But the basic mechanisms and ideas haven't changed. And it's not that in the end you don't want to learn all that detail if you're going to go into that field in a serious way. But I find when you're coming to the field from the outside for the first time, that detail can be very intimidating. And so those older textbooks have the basic ideas in them. They're shorter and they're very readable. And so I strongly recommend if you're approaching biological problems for the first time to go with an older textbook, something from 2000, 1990, and use that as your reference. And then if you want to dig into the field, you can go back and look at the more modern things. Otherwise, you really get lost with the trees, don't see the forest. So I recommend that. I like the Britain Essential Mathematical Biology textbook as a basic reference for a little bit of the math. We, in the other class that I teach, use this Herbert Sauro Systems Biology Introduction to Pathway Modeling, which I think is pretty good for these kinds of things. And there's some articles that are in the student materials folder that you could look at as well. There are online manuals for CompuCell. They're not perfect. If somebody wants to help us improve those manuals and get some extra points for doing that, I would love to have more demos in the CompuCell package. I would love to have more examples in our manuals. If people want to add to the manuals, we can show you how to do that. If you catch mistakes in the manuals, that would be great to help us out too. Okay, we have these quick reference guides. I handed out a Python introduction. If people want, I have more copies of the Python introduction here. That's just a generic Python course. It's not custom for here. There is a CompuCell quick reference guide, which is available online. For subcellular model, there's a tellurium guide, which I didn't hand out today. And so I encourage people to use those. I personally like to have those printed out because I don't like to have to keep flipping back and forth on the screen between the things, but it's a good case. If people want to know a little bit about what goes on under the hood in CompuCell, there's a paper that I wrote quite a while ago, 10 years or so ago, called Magnetization of Morphogenesis, Brief History of the Glazier model, which talks about how these things work. If you've had some physics background, that will be great. If you haven't ever had any physics, it may be not the most accessible thing in the world. You don't need to know it to be able to do the things we're going to be doing in the class, but it can help. There's this nice, if you've never done any mathematical modeling before, there's a nice paper from a year or two ago in PLOS Computational Biology by a group of Canadian graduate students called 10 Simple Rules for Tackling Your First Mathematical Model, a guide for graduate students by graduate students. We should probably write something similar for CompuCell. The handout that I gave was an attempt to do that, but somebody wanted to finish that paper, that wouldn't be a bad thing either. But this paper can give you some guidance if you've never done biological modeling. So those are some resources that you could do. As I say, nobody comes to a class like this, or almost nobody comes to a class like this, unless they happen to be a PhD student of mine or Paul's, pretty much, having all the background they need. And so, as Hayden has talked about, Hayden is an expert in science and engineering education. So he's a great person to have on board here. Talks about cognitive overload, which is, it's a little bit more difficult to try to learn a lot of different things at the same time than to dig in and try to learn one thing at a time. And I recognize that. And for this class, you'll need to know a little bit of biology, a little bit of mathematics, a little bit of programming, a little bit of physics, a little bit of chemistry. You're not going to need a huge amount of any one of them, but you'll need a little bit of all of them. And so I encourage people to think about where their gaps may be and work to try to fill those in, not in a frantic way or a dramatic way, but just gradually identify the things that are more or less familiar to you and fill them in. And again, working in teams is a way of helping each other do that. So those are some things that we could use. If people find material that they think are good as references, background education, please send them to me. We'll add them to the course materials and make sure that at least the subsequent versions of this course take advantage of that. This course will be given probably again in the summer as that intensive summer course. So almost anything you put in, help make this better now, will get used for 50, 60 people over the summer. Okay, one thing that I probably don't, since you're already here, I don't need to sell the course, perhaps. Maybe one or two people are shopping for courses, in which case, and this would apply as much to Paul's course or my course, which is that one of the things that we're very aware of today, or at least maybe we were until March of last year, was that there was an enormous demand for people in data science and artificial intelligence machine learning. Nowadays, with layoffs at Google and so on, maybe it's a little bit less that way, although they're still hiring people. But if I look at the number of people, say at IU, who are studying machine learning compared to the number of people studying this kind of thing, it's probably a 50-to-1 ratio. And and learning data science and artificial intelligence is great, and I strongly encourage everybody to learn something important, part of the modern world. But there is a real shortage of people with mechanistic understanding of how biology works and the ability to turn that into computing. Yeah, the U.S. Food and Drug Administration for new drugs now requires quantitative models of how those drugs are distributed in the body and how they work. That means basically all pharma companies are hiring people in this area. It's a little bit more the kinds of things I teach in the fall course or the spring course. But Paul Macklin's student Yaffe went to work for pharma. Giuliano, who'll be the TA, is probably going to go work for Lilly. There's a huge demand for people with this kind of skill set. And so if you want to go into industry, learning this kind of material gives you a bit of a differentiation from people who might have the classic computing, and maybe gives you access to not better jobs, but slightly different jobs, maybe a little bit more interesting. And there have been, again, of course, venture capital has dried up a little bit in some of these areas in the past year. But there are a couple of companies in this general area, like HeartFlow, Applied Biomath, and Nova Discovery. This slide probably needs to be updated because I haven't checked if they're still hiring as of today. 6 months ago, when we taught the summer course, they were all recruiting at pretty high levels. There's also the Embryo Institute at Purdue University that is a NSF-funded PhD program in this field. We've sent some students from here to there who've been doing pretty well. And so there really is a shortage of people with this kind of qualification, whether that's at the master's level or the PhD level. And so that's something to think about. So why are we here? And this is a slide that people may have seen from other lectures I've given. And fundamentally, there's a question, which is why do we need computational biology, the kind that we're talking about, computational bioengineering? And if we're here as engineers, and this course is listed as an engineering course, we start out by noticing that almost every thing that we deal with in the modern world that's built is designed by computer. It's monitored by computer. It's diagnosed by computer. And so if you're an airplane engine, a computer itself, communication system, a modern automobile, these are all designed by computer-aided design systems. They are all monitored, they're diagnosed, and they're repaired by computer design. Medical systems are just about the only things where we don't actually use mechanistic models to predict health state, to determine how to treat patients, how to maintain health, and so on. And the reason for that is fundamentally that engineered systems, we have blueprints. And so we can relatively, not easily, but it's at least conceptually straightforward to build models of engineered systems. There's still a lot of scientific uncertainty about how biology works. And biological systems are much more complex than even the most complex human engineered systems. So if we are going to be able to do design, the first thing we have to be able to do is prediction. Prediction is not sufficient to do design. Design is harder than prediction, but prediction is necessary. This class will focus on doing prediction. Sometimes the terminology for that is digital twins. Sometimes it's called virtual tissues. There are some technical issues which we could talk about offline about the difference. But one of the big things that I think is going to go forward in the next 10 or 15 years is going to be the question about, can we in fact predict changes in human health state? Whether you need the kinds of models that we're going to develop in this class, whether you use machine learning and database methods or something else, It remains to be seen. The national academies are doing a big study on medical digital twins right at the moment. I'm going to be speaking briefly at a meeting at the end of the month on that for the national academies. And I've been leading a discussion forum online for the past 2 years about this for the National Institutes of Health. And if people are interested in talking about that, I'm happy to do it. I think if I were trying to decide sort of what my future would be in the area of computational biology, I'd put myself somewhere in that field of what's called generically digital twins, medical digital twins. There are a lot of other things one could do, but that I think is really pretty important and pretty promising. One of the things you face when you think about medical digital twins is that it's more like, not like building a digital twin of an airplane engine where there really is a single blueprint and that engine is supposed to work in a particular way, but more like building a digital twin of a city where things are changing. There are things we don't know about. People come in from outside. There could be accidents. There could be tornadoes, or like weather forecasting. And so there are a lot of quite interesting issues in digital twins. I'd love to give a course specifically on digital twin design at some point, but that's something we can talk about. But if we're going to make predictions, we have to understand some basics of the science of how things work. People who do machine learning might argue that you don't need to understand things to do prediction. My friend Gary Ann, who's a surgeon and also a biomedical researcher, points out that there are certain things you can't do with machine learning. Machine learning depends on the training sets that you use. And so one thing that machine learning is not good at is dealing with what are called counterfactuals. How do you deal with situations that have never happened before? By definition, if it's never happened before, it's not in your training set. And machine learning methods tend to do really poorly in those situations. The other way machine learning tends to fail is something very connected to how biology is organized, which is that machine learning is great for what's called linear extrapolation. If the same input always gives the same output, machine learning does really well. If there are a lot of layers of organization between the control and the output, then machine learning does less well. And this is a problem one faces in medicine in particular, because most of our medical treatments are drugs. Drugs work at the level of molecules inside of cells. But what we care about is not what happens inside a single cell. What we care about is what happens to your tissues, your organs, and you as a whole organism. And there are molecules that can be lethal to individual cells that don't do any harm to you as a whole body. And there can be molecules that don't seem to do very much within individual cells, but disorganize tissues in a way that can be lethal, and vice versa. One of the fundamental principles of engineering is that you want to have the level at which you do control be as close as possible to the level at which you make measurements, and as close as possible to the level at which the output you care about happens. And in medicine, that's usually not true. Surgery would be an exception. You cut something out, you cut it out. But chemical treatments, drugs operate very far from the level that you care about. And the measurements that we can make in the clinic also tend to be at the whole body level. We don't have tricorders which can go into your body and measure every cell in your body in real time. If we did, we wouldn't know what to do with the information at the moment, but that's an issue. And so there are some fundamental principles that have to be thought about there. And there's missing science. We don't really understand how organisms develop. How do you go from a single fertilized egg to the structure that you have? One thing that we really do know is that there are no blueprints in biology. Systems organize themselves. The principles of organization in biology are fundamentally different from those of engineered systems. And that means that the problem of design in biology is quite different and more complex than that in human engineered system. Something that you may not think about so much, but that is really critical, is how do organisms maintain themselves. A table, if we leave it alone, will stay a table. In your body, if we left everything alone, you would die because you need a continuous input of nutrients, oxygen, energy, and you have to get rid of it. And so your bodies are dynamic. Cells are continually dying, being reborn. Tissues have to be restructured. And maintaining structures without a blueprint is actually pretty difficult. We have, because of that, what are called developmental diseases. The mechanisms that happen to be responsible for development and maintenance of tissues have to be regulated in some way. When that regulation is perturbed, you have failures of organization. You get disorganization. You could think of a traffic jam as a classic example of an emergent problem at the systems level. And you could actually, as a metaphor, you could ask the question, if I change the speed limit on 3rd Street, what happens to traffic flow in Bloomington? It's not obvious. You might say, well, I make people drive, if people drive faster on 3rd Street, then traffic will flow better throughout the city. But there are plenty of studies that show in many cases that if you make things go faster in one place, you actually create bottlenecks somewhere else. That's an example of this issue, that having the level of control you have, changing the speed limit, doesn't directly control the thing you care about, which is how does traffic flow at the level of the whole system. And so those kinds of situations are ones that we have to think about carefully. There's scientific uncertainty in biology. There's still things that we don't understand about how systems work. And those are all issues that we really need to address. Paul and I have worked quite a bit over the past 2 years on infectious disease modeling. This class was taught around infectious disease modeling in 2020, 2021. Then students said, we're tired of infectious diseases. We want to go back to thinking about development and cancer. So we've gone back to development and cancer. But if people are interested in modeling infectious diseases, we have lots of slides and examples of it. And then potentially, how do you do bioengineering? Can we use living systems to build things, not natural things, which is the first part, but rather artificial structure? And that's quite interesting in its own right. There was a period when tissue printing was all the rage. We were going to put cells down in particular patterns and build artificial organs. It hasn't really worked very well so far. You could put the cells down in a pattern, but what they do afterwards is not necessarily what you want. So being able to predict that would be nice. One of the key things that we're going to face in biology is that there's a complex interplay between physical systems, things have to move, things have to exert forces, and biochemical control. We're also going to face the fact that there's a lot of noise. Biological systems are intrinsically very random, and we still don't understand to some extent how you get repeatable large-scale results in the presence of very large-scale, high-level noise at low scales. Some of the projects that we've worked on over the years as research projects using these kinds of methodologies. Why, when people age, do the blood vessels that normally stay behind the retina and keep it oxygenated and fed sometimes invade the retina and tear it to shreds? Could we improve treatments for diabetic diseases of the retina? Which are not very satisfying at the moment. What causes some people to have something called polycystic kidney disease, where the kidneys never stop growing? You wind up dying in your 50s with kidneys the size of watermelons. I had a long-term collaborator who was a surgeon who worked on that. Why does an analgesic like acetaminophen, Tylenol, work fine in some people and cause liver failure and kill you in other cases? Or how do we use an antiviral drug to control viral infection. So we're going to be talking about virtual tissues. If we look at biology, especially developmental biology, these are all videos of living organisms. The one on the top right is a famous movie from 2008 by Keller and collaborators. In this case, they used a molecular label to mark the nuclei of cells. And then they used a technique called scanning planar imaging microscopy, SPIM, to do 3D imaging of an entire embryo while it was alive. And this happens to be zebrafish. Maria Bondison here at IU is a zebrafish lab. If you want to take her class, she can teach you how to do an experiment like that. And so here they were able to actually track the nucleus of every cell in that embryo as it was developing. It's a beautiful movie, but what do you do with that information? How do you understand it? Here is a little bit more detailed picture of a zebrafish. That was actually taken by Maria Bondison and some of her collaborators before she moved to India, showing the blood vessels that are going to go between the segments of the body, between essentially the ribs of the organism growing as the animal's alive. Before COVID, I used to do wet lab biology as well. I worked mostly in tick. It's not my movie, unfortunately, but this is a video of a chicken embryo developing. Great thing about zebrafish or tick, unlike human embryos, is that you can put them in a dish and they stay alive and you can watch them. A little harder to do that with a mouse or a human being. And here you're seeing that segmental structure forming in the middle of the body. And that's something we've worked on in our lab, both experimentally and computationally quite a bit. The top one is about the branching of the lung. And what you'll see is there's a lot of dynamics. Cells are moving around, they're changing shape, they're exerting forces, there are flows in the tissue. There's a lot of physics going on. It's not just signaling. And so if you're doing pure bioinformatics and trying to understand what molecules are present in a particular cell, you could know everything about what's happening inside one cell there. But if you don't know what that context and those changes are, you're not going to understand how the system works. And it's that kind of multi-scale organization that's really going to be the focus of this class. And so what we're going to do is we might call a middle out approach, where we start out by thinking about cells, because they're the active agents in biology. They're the only things that are really alive and active, and what they do and how they interact with it. They have inside of them molecular control signals, gene regulatory networks, metabolic networks, signaling networks. Sometimes we'll model those in detail, sometimes we won't. Sometimes the control is simple. And then the cells and their behaviors play out at larger scales. In this case, this is a simulation of that process in the tick called Angiogenesis, which is giving rise to the segments that are going to eventually form the periodic structure of the rib cage and the spine. In the middle, we have long-range chemical signals that are changing in the embryo. In the right hand, let me play that again. In the right hand, we have a mapping of a particular chemical signal inside individual cells. There happen to be oscillatory chemicals inside the cell. So there's a gene regulatory network that's modeled there. On the left, the gene regulation and those external signals lead to differentiation of the cells, change of the mechanical properties, and that leads to rearrangement of the tissue, leading to the formation of these segments. And so that would be the kind of example of a tissue model that we might hope to build in a class like this. Now, as I say, somebody said that they were interested in bacteria. You don't have to have your cells represent individual components of a multicellular organism. Those cells could be bacteria facing food in the environment, sending signals to each other in their environment, swimming in response to the environment as well. When we go to more biomedical applications, we'll often put a model of this kind inside of a transport model, a non-spatial transport model of the blood and the lymph and the whole body, what are called PBPK models. Those are typically used when we want to understand something like drug dosing. If you imagine you take a pill, you want to know what happens. What's the availability of that molecule in different tissues? What are its side effects and so on? So that's possible too. You can put higher levels of organization. So. Okay, sorry. So sorry. It should be there was a slip that slippage there. It should say it's supposed to say PDE, partial differential equations. I think there was a formatting issue when I created this slide. And so, yes, I will try to define terms. These slides often are borrowed or reused from other things. And so if there's a term that you don't recognize, partial differential equations in this case really represents diffusion. How are chemicals secreted by cells? How do they move around in the extracellular environment? And how are they taken up by cells? ODEs, ordinary differential equations, and that's a way of mathematically representing chemical signals within inside of cells. So these are going to be, we're talking about mechanistic models here. They're dynamic, they're agent-based, if people have heard that terminology before, and they're multi-scale. And our interest is fundamentally going to be on emergent phenomena. We're going to have objects, typically cells, although those objects could be individuals in space or components of cells as well, that are going to interact. And we want to know what happens when we put those objects into an environment. What do they do? What do their large-scale organization do? It's mechanism-driven. These aren't data-driven models. That is, we have to have an idea about the physics or the mathematics or the biology of the system that we're going to be modeling. They're additive, which is the opposite of data science. Data science, you start with huge amounts of data and you design filters to gradually select the things you care about. Here, we start out with a blank slate with nothing in it. Nothing exists unless we assert it. We're going to be focusing on cells to organisms, although again, you can build higher scale models as well. And it's going to be based on understanding basic properties of what those agents do. How are cells born? How do they die? What forces do they exert? How do they move? How do they change state? How do they change type? And the outputs of our simulation are going to be like what we saw there, which are going to be movies. And one of the things that we're going to have to face is how do we analyze those movies to come up with meaningful results? It's not easy to do. There are, in a living organism, there are many more cells than we could possibly model. Even the biggest and fastest computer simulators can't simulate more than about 1 million cells, 10 million cells at once. In this class, we'll be simulating thousands of cells at a time. There are lots of different kinds of cells. No two cells of the same type are the same. There are lots of parameters, a lot of which we don't know. And we don't know all of the detail of biological processes. There are a lot of unknowns. And so one of the hardest things that we have to do in simulation is to decide what we're going to leave out. Modern bioinformatics loves to have long lists of every molecular component possible, which is great. But as I like to say, if I give you a list of all the parts in your car and ask you, now, given the list of parts in your car, tell me how your car is to drive, you can't do it, because the parts list doesn't specify how the system works. And so this is a fundamental problem with contemporary molecular biology, that there's been a huge focus, beginning to change now, on creating parts lists and not so much on how systems work. Please. Yes, that's exactly what we tend to do. We'll build the simplest possible model of the system. And then we'll ask the question, is that sufficient to explain what we want? If it isn't, then we begin to add additional control, additional complexity. How far we'll get with that in a semester is another question. Sometimes these things can be many-year projects. But at least in principle, that's a very good way to approach things. And that's also true actually in data science and machine learning. If you like TensorFlow, the Google tutorials on TensorFlow I think are really, really good. And they have a really wonderful one on using machine learning to do time series prediction. They have these weather measurements. And they start out by saying, well, what if you start out by just assuming that the next time step is the same temperature as the current time step? Winds up that actually does pretty well because temperature doesn't change that fast. And then the next thing is, well, what if you just do linear extrapolation? The change in temperature every hour is the same as the last one. Well, that does pretty well too. Unless the temperature is turning around between day and night, it does pretty well. And so it winds up that these very simple kinds of extrapolatory rules for a lot of things actually do pretty well, but they don't give you understanding. And so I think that TensorFlow tutorial is great because it asks the question, suppose that you have a model that's, quote, 98% predictive. You know, some AI model is 98% predictive. If your assumption was just nothing changes, you find out you were 85% predictive already. And so that means that actually doing better than the naive things is pretty hard. Here, we're exploring what the capabilities of particular mechanisms are biologically. And so we're coming from a slightly different perspective. But how you organize, for example, how bacteria organize to build biofilms. It's a classic kind of simulation that's done with these modeling methodologies. How quorum sensing works in biology. They're often done. And so there are a lot of other issues too. How do we compare experiments and simulations? Not so easy. Missing science. Range of length scales. It's hard. There's some nice there are also a lot of different ways of building simulations. How you represent cells and tissues is different. There are a lot of different methodologies. Some of them are faster than others. Some of them are more detailed than others. Each one has its own set of advantages and disadvantages. People taking Paul's course and my course, Paul's PhysiCell can simulate more cells at a time. It's faster, but there's no spatial detail. In this class, each cell has more spatial detail. If you can treat your cells as point particles, PhysiCell is great. If the cells are extended objects with complex structures, then that's not so great. And so the trade-offs that you make are going to be things that you have to address. Here are some examples of different kinds of simulation. These are what are called the center models. These are based all on molecular dynamic simulations. That picture of Paul's is rather old. He has fancier ones now. But each particle is essentially a point with an interaction range. As I say, this comes out of molecular dynamics. These are very fast models. You can do lots of cells at a time. And there are lots of different packages, PhysiCell, Case from Oxford, Biocellian from Pacific Northwest Labs, all use this kind of center model approach. And you can do an awful lot of neat things with that. But sometimes you want to know how cells shape matter. And there are lots of different ways of doing that. CompuCell is one. There are people who use sort of solids modeling packages, something like Abacus, to build very detailed biomechanical models of cells. Those are fantastic, but also incredibly expensive computationally. There are also things called vertex models that we won't talk about so much in this class, that are useful for particular things that can be very beautiful. And so there are lots of different ways of representing cells. There isn't a right or a wrong way to do it. There's differences, levels of convenience, depending on what you're trying to do. Ideally, you'd like to have a way of specifying a model that talked about the biology that would allow you to use any of those methodologies to solve the problem numerically. Unfortunately, that isn't available. There is no standard way of specifying these multi-scale models. As I say, we're hoping to be able to build some joint specification with Paul and PhysiCell in the next year or so. And we're working with a group in Dresden also trying to do the same thing with their modeling. And one of the things you'll probably find out, especially if you're taking Paul's course and our course, this course simultaneously, is why it's not so easy to write a joint specification. I'd love to be able to do it. CompuCell, the software package we're going to use, exists in two components. There is a model editor called Twinit++, that is a smart editor that has a lot of features that make it easier to build models and debug them. And then there's something called Player, where you actually run the simulations and visualize them. You can pull this on your desktop. And I certainly don't discourage people from doing that. If there's a problem getting the thing to install on your desktop, there is a version online through nanoHUB that we could use in class. And so the online one is a little bit older version of the code. Today, assuming that people don't have it, how many people, if anybody, has anybody installed CompuCell on their computer? 2, 3, 4. Did it work? So if you have a Windows machine, it should just work. If you have a Linux machine, then you're probably either going to have to go talk to Maciek and TJ, or we'll have to use nanoHUB version. So the main reason that we have the online version is so that if somebody has an exotic version of Linux or has the very brand new Macs, which are unfriendly, you can still work without getting too distracted by installing stuff for forever. Okay, but we have the default for this course, because we're not building core code, is that if it won't work on your local machine, we use the online version. And I don't mean to discourage you. It's just great. Hey, if it's launching, that's great. I'm not going to complain. But there is a backup, which is that if there's a problem, we have an online version that works. The online version has some annoyances, but it's for example, when we teach that summer course and we just have a week, that looks great. When we teach the summer course and we just have a week to do it, we don't have time to debug 60 people's installs. So do that. Okay. CompuCell has a lot of built-in concepts. It has the idea of a field, which could be a chemical field or electrical field or something else, where there's a concentration at each point in space. And its basic ideas are diffusion, decay, secretion, absorption, and to some extent transport. There's a concept of a cell or a generalized cell, which is just a volume of material. It could represent a biological cell. It could represent a structure like a nucleus or organelle. It could represent bacteria, but it could also represent just a little bit of fluid or a patch of cell membrane. And in those have concepts like volume, polarity, surface area, viscosity, growth, division, depth. And they can interact with chemical fields. Cells could move up and down gradients of chemicals. They can absorb and secrete chemicals. It winds up that the fundamental concept of cells in CompuCell is soft. It's very plastic, fluid-like. Cells build tight junctional structures like adhesion junctions between themselves. They have fiber structures like filapodia, which they use to anchor onto things. And so, and you also have solid materials like elastic materials. And so CompuCell has a concept called, not very elegantly, focal point plasticity of CompuCell, or what typically called links, which allow you to build these kind of more solids-like behaviors. And then there's a concept of networks, a generalized concept of networks, which could be used to represent the regulatory networks inside of cells, transport networks at the whole body level, and so on. So those four are fundamental concepts of fields, generalized cells, links, and dynamical networks are the things that we're going to be working with. Those are the objects that we're going to be building things out of for the next 16 weeks. So let's actually try doing a little bit of an exercise. Running CompuCell, some people have done it or not. So why don't we do this? I'm going to show these, and I can do this. I can do a split screen if we like. Let's see how it goes without a split screen to begin with. So the first thing I want to do is I want to run a simple demo that exists in the CompuCell package on vasculogenesis. This is based on modeling that was done by Roland Merks back here when he was a postdoctoral student many years ago, 15 years ago now. He's now a professor at Leiden, University of Leiden in the Netherlands. Goodness, it's more than 15 years ago, 17 years ago. Goodness, all right. And this is something that we can do together very quickly. We won't go into the details, it's just to get a sense of running CompuCell. The basic biology here is asking the question, how do the cells that form blood vessels organize? And blood vessels can form in two ways. You can have an existing blood vessel that has sprouts that branch off from the existing blood vessel. But in peripheral tissue, like in the limbs, the things that are going to form the blood vessels are initially distributed a bit like raisins in bread, and they come together and aggregate to organize blood vessels. And those seem to be very, very different ways of building a blood vessel. And the question that we asked all these years ago was, do you need fundamentally different mechanisms for those two things to work? Or is it really just a question of the same mechanism starting from different initial conditions? And there was an old hypothesis that there was a growth factor, vascular endothelial growth factor A, VEGF-A, that cells secreted and responded to. Cells secrete the chemical, and they also move towards higher concentrations of the chemical, autocrine, paracrine chemataxin, technically. And there were some experiments by Gamba and Sariti going back 20 years now that seemed to suggest that that was possible. And we were involved with some experimentalists who showed that there were some other things going on about how cells stuck together that seemed to be very important. That cells, endothelial cells, when they were had free surfaces, were able to respond to external chemicals, but when they were touching each other, they didn't respond. And so, based on those hypotheses, we built a little simulation of this. These cells are secreting a chemical. They are moving up to higher concentrations of the chemical. In the top right, there is a movie of the cells in a dish doing the same thing, living cells, organizing a vascular network. Actually, one of my students did quite a bit of experiments on that. That's not our movie, but he did a lot of experiments on things. He has a patent on ways of doing that better in a dish, experimental. Here, what we see are two simulations. The one on the right, the cells secrete the chemical, they respond to the chemical, but the surfaces where the cells touch each other don't see the chemical. On the left, all the surfaces of the cell see the chemical. The only difference is that all the surfaces of the cell see the chemical are only the free one, And you see in one case you get blobs of cells. In the other case you get that vascular tree. So let's run that simulation together. If you have CompuCell running on your computer, that's fine. If you don't have CompuCell running on your computer, it probably is takes a little bit of time to download it. And so I would recommend going to nanoHUB. I encourage everybody to set up a nanoHUB account. It only takes a few minutes to do it. So I'd like people either to launch CompuCell locally or go to nanoHUB. This QR code will take you to CompuCell on nanoHUB if you do that. So, yes. I'm sorry? We will take, let's see. That's actually good. I usually take a 5-minute break halfway through. And so do remind me of that. So actually, why don't we take a break till 6:20? And if people want to use that break time to get CompuCell running or get onto nanoHUB, that's fine too. So why don't we take a break till 6:20? Thanks. Do remind me of that. Thank you very much. Now, 3 hours with no break is too long. In the pre-COVID days, I almost always brought cookies or chocolate or something to help get through these evenings, but these days, not so much. Are there any questions from our online people about this? Does anybody need help finding the nanoHUB CompuCell or getting it downloaded? Again, if you have a PC, Windows machine, the download and install is somewhat slow. The new installers are a little bit on the slow side compared to the old ones, but they should just work. No promises. If you have some complex set of Python libraries on your computer, you may have an issue. The installers went from pip to conda, which was supposed to make things more robust, and it did not necessarily make things more robust. And I'm afraid I'm not the person to debug that. If there are problems with installation, we have to go to Martek and teach. There's always a lot of diversity, as I say, in this class here, there's a lot of diversity in terms of the level of seniority of the students, whether the students are computing, biology, sometimes mathematics, physics. We've had students from chemistry, psychology take the class. So very great diversity of background. Mathematical biology. Right. So that biophysics textbook, the Phillips biophysics textbook, I think is extremely beautiful. He's at Caltech, Rob Phillips. His interests are primarily in intracellular machinery. He's very interested in molecular motors. And so that textbook will talk about how myosin motors work. He does have good chapters on transport, blood flow. The sections on developmental biology and patterning are not the strongest in that textbook. I shouldn't be saying that probably online because he's a friend, but I think he would probably be open to saying that that's not the specialization of the textbook. But there's biophysics textbooks tend to be very heavy on statistical mechanics. Yeah. And that one too. I mean, the other book that's famous as a biophysics textbook is by Phil Nelson from Harvard. Well, I guess he's actually, is he at Harvard still or is he at Penn? Yeah. And that's even more of a statistical mechanics textbook. What I like about Philip's textbook is he wrote that with a number of experimental biologists. And so a lot of biophysics textbooks tend to be, here's some physics idea, how do we use it? Here's a physics idea, how do we use it? Philip's book is very biological. It says, here's a biological problem, how do we solve this problem? And ultimately, it covers the same material, but it covers it starting with what are the biological problems rather than what do we do with the methodology. The math book, the essential mathematics book is more on ODE methodologies. It's really a dynamical systems textbook. It covers ideas of stability, bifurcations, control, and it does it in a pretty elementary way. It's designed as an undergraduate textbook. Has pretty good exercises. Mainly OD models. Yeah, they do a little bit of spatial modeling, but not a lot. The standard mathematical biology textbook is by Murray, called Mathematical Biology, in 2 volumes, the green cover. It's a Springer green cover book. I think the Springer textbooks are available to people here for free. I think IU used to have a license, a site license for all the Springer textbooks. I don't know if that's still good. And Murray in volume 2 has some very nice very nice chapters on patterning, spatial patterning, what are called reaction diffusion equations, which you can do in CompuCell. And not so much on agent-based modeling because there's not a lot of analytics you can do. Murray is very much analytics person. Just MURRAY, mathematical biology. It's such a good textbook that basically nobody has written another one since he published that. In the days when I would teach mathematics courses, which I did, physics and mathematics courses, I taught biophysics for many years, and I used the Phillips textbook for that. And then for math bio, I would teach it out of Murray. In this class, yeah, that's what I thought. I think it's available to people here. Yeah. Again, people coming here in intelligent systems engineering, people have diverse backgrounds. I don't want anybody to be excluded if they haven't had a lot of mathematics or physics. And so I'm happy to work with people and digging into more of those things. But in the classes, I'll try to minimize that because just to make things a little bit more accessible. Yeah, absolutely. I'm happy to do that. I mean, the level of resources that are available is large. As I say, the one thing we don't really have is a textbook on this specific methodology, sort of an irony.
Okay, what's this one about?
Right, that's the Italian book on Tott's model is as close as we get, but it's a little bit old and it wasn't written exactly as a textbook. in the sense that it covers a lot of good material, but it's not organized it's not organized pedagogically with exercises, for example. It doesn't have a problem set, which would be helpful.
So, like,
So we have lots of publications that talk about how the things work, but it's not, again, they're not pedagogical. I'd say our online manuals are probably the closest thing that we have. There's a quick start guide, which is a little bit out of date, which tries to take people through this material. In a sense, the stack of slide decks that we have here really need to be turned into a textbook. So that's one of those things that needs to get done. Okay. And again, I'm happy to talk more about these at any time. So how are people doing in terms of getting CompuCell up? Is everybody back, first of all? What people will find out online, and this isn't going to work perhaps as well as it could. I often will use when we're mostly are all online. I'll use this exercise completion poll, but that doesn't work. So that's great when everybody's online, because that way I can see how people are doing. If people are in the room, I can just look and ask you how you're doing. But people are saying they're finished with the exercise. Okay. So that's how we keep things synchronized. So not a very sophisticated way to do synchronization, but it works for online work. Okay, good. Everybody's back. That's what we need to know.
So sorry, but where should we? So I'm using a Mac, and I've had some trouble. I'm just running it on nanoHUB, but I was having some trouble actually installing it earlier.
Yeah, if you're on a Mac, life is not going to be trivially easy. So why don't you stick to nanoHUB for the moment? And when Giuliano gets back, you could ask him for help with the Mac install.
Awesome. Sounds great.
You can also write to TJ. But TJ, I don't want to go into online why there's an issue with the Mac installs. But we'll do our best. But for the moment, the nanoHUB will work. People who've used nanoHUB before will know that the biggest problem with nanoHUB is transferring files between nanoHUB and your local machine, which is not the end of the world, but isn't that great. People who took the fall semester are used to this. There's going to be one additional annoyance, which I'll introduce you to with the nanoHUB version of CompuCell3D. But so let's go forward here. So if you're going to, assuming you're going to go to nanoHUB, how many people in the room are running locally, and how many people are going to run online? Let me just ask a different question. How many people here want to run on the nanoHUB? Okay. So nanoHUB people, online people, is there anybody who, how many people, could you use a thumbs up if you're using nanoHUB online, please? 1, 2. Okay. Local. Okay. 3 local. Anybody else? Okay. Okay. So for people who are using nanoHUB, how many of you have gotten into nanoHUB so far? 1, 2, everybody. Is there anybody who is not able to get to nanoHUB? Because I can skip the next couple of slides if you've got that. Online people, is there anybody who is stuck and are not able to get into nanoHUB? Gabriel says he won't use nanoHUB willingly. That's good. All right. All right. I won't. You just said that on the record. I won't report that to the nanoHUB executive. But, we are actually trying to do Jupyter Notebook version of CompuCell. And when that happens, then you'll be able to use something like Colab to run it. So you'll have more flexibility in terms of how to execute it in the cloud environment. But that's not deployed yet. We actually had an undergraduate student from a community college who did a lot of that for us last year. We had a REU research for undergraduate program. It was great work with Trinity doing that. Okay, so people have gotten onto that. Does anybody need help getting into nanoHUB? It's going to be a boring hour and a half if you can't get it to either your local machine or nanoHUB. So please let me know. Don't be shy. Okay, I'm assuming this means everybody is either in nanoHUB. Everybody's in nanoHUB? Good. Okay, so if you're in nanoHUB, there's something called nanoHUB dashboard. You can hover over the top right-hand corner where you say logged in, and you go to dashboard. Okay, and if you're there, you could launch CompuCell Player. So what you need to do there, and I could do this, sometimes I'll do this split screen and do this, type this as we go, but if people are already ahead of me, I don't want to take too much time on it. If you go to dashboard, you will see a window called My Tools. You could search CompuCell3D V4 main tool, and that will take you there directly, or you could search in My Tools CompuCell3D V4. And when you do that, under all tools, it'll pull up CompuCell3D V4, and the little heart is a bookmark. So if you click the heart, then it's available to you. So I'll give people a minute or two to get that to work. My hesitation about doing this with a split screen is mainly that if I do that, then for people in the room, this gets pretty small. Some of these classrooms have very large screens, so it's easy to see. Here, if I split the screen, it gets. We'll be doing split screen things later. But okay, so did everybody find CompuCell? Okay, there is an old version of CompuCell, so make sure it says CompuCell3D V4, not just CompuCell. Okay. Everybody got it? How about our online people? Can I get a thumbs up for people who have CompuCell running? If you're running local, that's fine. Just give me a thumbs up. Okay, Mohammed, Nick, what about? Oh, you're online, but you're also on the. Okay, that's fine. Okay, so what about people in the room? Either way, that's fine. Everybody got it? Anybody stuck? No. Okay, so we're going to now run some demos. One thing we need are more demos for CompuCell. There are a variety of demos, which are small simulations introducing programming concepts in biology. Some of these are deployed as freestanding nanoHUB apps, but let's run a little angiogenesis simulation. As I mentioned, CompuCell consists of two tools, Player, which runs the simulations, and Twit, which is the model editor. These are really modeling tools. They're not models themselves. And so the first thing that we need to know is when you launch Player, which is what will happen when you start CompuCell on nanoHUB, is that the windowing will be a little bit odd. And so you have to do some resizing. So you'll have to be able to grab that bottom right-hand corner of the window and adjust it. Depending on how nanoHUB is feeling, it may take a little bit of effort to get that corner and pull it. Sometimes nanoHUB is a little recalcitrant about allowing you to adjust the screen size. Otherwise, you can use the bounding boxes in a normal way. It should be pretty straightforward. So if you're running locally, you just start CompuCell Player, And so everybody should have, whether you're in nanoHUB or locally, you should have something that looks like this. If you're running locally, you'll see this internal gray window. You won't see this X11 bounding box. That's a nanoHUB. I hesitate to say pizza. nanoHUB is wonderful because it's public service that anyone in the world could use. You could be anywhere in the world to get a nanoHUB account and use it. Get free computer time. It's great. But the flip side of that is there are some limitations in what you can do. And so in particular, we had to package CompuCell through an X11 interface. And looking at the people in this room, X11 is older than many people in this room. I happen to remember when X11 protocol was introduced. And it was sucky even back in 1985 when it was first introduced. It was already obsolete by then. Compared to, it was already behind open windows and other packages. But X11 is the one that's still around. So you get the pleasure of X11. And you'll discover one annoyance, which is X11 was introduced before the concept of a clipboard existed. And so you cannot cut and paste into an X11 window from other apps. So this is not going to be a huge thing, but if I'm teaching this course and I put text in the chat, you have to retype it. You can't, or upload it. You can't cut and paste it into your simulator. That's one of those. Okay. Everybody have CompuCell running. You can play around with the bounding boxes. Okay, so what we're going to do next is we're going to open a simulation file. And so if you go to the top left-hand corner of CompuCell Player, you'll see a pull-down menu called File. Pull it down. The first line will say Open Simulation File. And then if you are on your local machine, you will navigate to the CompuCell directory, and you will find the file called CompuCell3D demo. If you are on nanoHUB, you will find that there are two directories which contain CompuCell CC3D demos. One of which is the root directory for the software itself, which you don't have edit access to. And the other one of which is your username, whatever you are, and then you'll find CompuCell CC3D demos. And so, for example, here, this was this was written by Giuliano. So you'll see if you do open simulation, computer, J. Ferrari, which is his account on nanoHUB. And within that, you'll find CC3D demos. So if you're on nanoHUB, you have to make sure you first click on your user account and then open it. Because otherwise the thing will fail because it will try to write to a directory you don't have write access to. If you're on a local machine, that will not be an issue. So I'll give everybody a minute to do that. If anybody has trouble finding it, let me know. Well, okay, I haven't gotten to the model name yet. So you go to click open simulation file, find, you should get something that says computer, find the folder that has your username, whatever it is in it, open that, and it'll pull up a window called CompuCell CC3D demos, okay? Open that. Inside of that, there are a lot of directory hierarchies. There should be something called demos, /demos. Inside of that is something called book chapter demos. And inside of that is something called angiogenesis. And inside of that is a file called angiogenesis.cc3d. And I realize this is almost a Microsoft level of nesting of directories, which is not the most friendly thing in the world. So give people a minute to find that. Is everybody finding it? Not finding it? How about our online people? Jonathan, you got it. Anybody else? Carmen, Aiden? You got it. Got it. Okay. Great. Thank you. Oh, good. Great. How about in the room? Everybody got it? Anybody stuck? We're going to do this a lot, so we'll take the time we need now to make sure. Were you able to get it to work? Great. Everybody got it? Okay, good. If you're having trouble, don't be too shy about it. If you're having trouble, probably everybody else is. Let me switch to split screen here, and I'll run it locally as well. Uh, are remote people able to see the screen all right now? Is that working for people? Okay, so here's player, and what you'll see on player are a couple of buttons. The leftmost button, like a play button in an old cassette recorder or CD player, is play. That runs the simulation. The one that looks like the old record button with a circle on it is single step, the simulation. There's a pause button, which is currently grayed out, one to the right. And then there is a stop button that terminates simulation. People have a tendency to hit stop. I strongly recommend don't hit stop unless you really want to stop, because if you hit pause, you can always abort the simulation. But if you hit stop, you have to start from the beginning again. So I strongly recommend using the pause button when possible. So here's what we were going to do. I hit File, Open Simulation, click on my name. And here I have I've got a lot of demos and things in my directory because I've been doing this for a long time. I'll go to CompuCell3D demos, book chapter, angiogenesis. Okay. You're stuck? Oh, blind. Me too. So that was, as I say, the problem with split screen is that things get pretty small. Sometimes we teach in that, I taught this in that classroom in front, which has that huge full wall screen, which is nice. The other thing that you could always do is you could always stream the Zoom to your laptop like you're doing. Of course, you have to switch screens to see things, but it's possible to do that. I've had some people who like to actually watch the YouTube, because you've got a lag. So, okay, is that okay? I'm going to hit open, and now I can start the simulation. I can start the simulation by hitting. I'm going to hit step here, and it'll launch the simulation. And I'm probably going to have to resize the window. And one of the things that sometimes happens one of the things that sometimes happens in the nanoHUB version of CompuCell is that the simulation will disappear on you. So you may have to do some looking around to find the simulation. So hit play, and it should create a, there we go. Create a simulation window. Do people get that to work? Anybody stuck with that? Yes. Okay, that's fine. That looks easy. I mean, it's not exactly, okay, so let me talk a little bit about one of the exercises we're going to be trying to do now is just to get familiar with how to manipulate visualization in CompuCell. So you're running locally or are you running on nanoHUB? So the visualization is a tiny bit different on two. One feature that exists in PCs and nanoHUB, but not on Macs, which is very convenient, and this is not our fault, this is a Mac problem, is if you go under the Windows menu here, the second from the top called Tile will lay out your screen. And so if you lose your simulation, you can hit Tile and it should find it for you. Okay. The other thing, if you get things in a strange state, if you hit the letter, you hover over the screen and you hit the letter R, it should reset the screen set. So does everybody have something that looks more or less like this at this point? If people don't, please don't be shy about asking. Okay. The other thing that you might want to do is this is actually a three-dimensional simulation. It's called CompuCell3D for a reason. And if we go up here to where it says XY, it's a little bit small, I can switch to 3D visualization. And boy, it's small, huh? And I can resize. In my case, I have to use two fingers to adjust the size. Sometimes depending on your mouse, you can scroll, or there may be a scroll button or something else to resize. So you should be able to see this in 3D. And I encourage people to play a little bit with the visualization to get familiar with that. You could also rotate. If you click left click and drag, you could rotate the visualization. Spreading your fingers will make it bigger. Pulling it back together, make it smaller. R should reset. Everybody got that? All right. And so now we could hit run. And you can run it. And you'll see things happen. You'll see things moving around. Does that work for people? Yes? Questions? R, the letter R. R stands for reset. So if you hover over the screen It's easy if you accidentally scroll off to the side or make things huge or tiny, you can lose your visualization. So R is supposed to reset it. Okay, were people able to get it to run? I guess I should be asking, since everybody seems to be get ahead of me, is everybody not able to get it to run? Good. So I would, if you want to start it over again, you hit stop and play. And on nanoHUB, the window will revert back to its old smaller size. So you'll have to resize it. On your local machine, it should not do that. And so you could explore, if you go to 2D, you can see a section through the tissue. 2D slice through it. If you go to 3D, you'll see it in 3D. And you'll see the cells moving around. And so take just a minute to play around a little bit with that and how to manipulate things. One thing, when you change the visualization, so you go from 2D to 3D, the simulation will pause and you'll have to hit play again to continue the simulation. All right. Now, no, it's not working for you. If you're while they're debugging, one of the things that you'll find when I give exercises in particular, I will often give a base exercise, and then there'll be sort of further extensions. So if you're having trouble or you need help, do the base exercise. If you're already there, instead of sitting and getting bored, you could continue with the extras. So we'll try to do that. Here, I would say one of the things that you've got as options here under visualization in this menu is you could turn on how the rendering has happened, where you show the surfaces of the cells and so on. That's something that you might want to do. Another thing that you could do, which is pretty simple but is useful, is if I go under Tools and I go to Configuration, you'll see something called CC3D Preferences, and you should pop up a window that says Cell Colors. And I can change, suppose I don't like the blue, I want my cells to be red. I click OK. And I can also control in 3D, I might want some things to be transparent so that I can see through the tissue. So if I have a layer of cells, I have to go see through them. I make them transparent. So I can control the colors that I'm going to use. One thing that can happen in nanoHUB, which can be a bit of an annoyance, is occasionally the cells will disappear. It's a bug that was fixed in the newer release, but is a problem in the old release. If that's the case, and everything disappears on you, right? Can you do that for me? That's not right. If everything disappears on you, you can go here, hit this button here, create a new graphics window, and then you should be able to see what's going on. So we can run and play with that. So everybody can try changing the cell colors. Here, go to cell colors. I'll make the cells here endothelial cells. I'm going to make them red. Okay, okay. And now let's make this all. That's not a big deal, but if you have 15 different kinds of cells, it's nice to be able to control what the color is. You can also control the boundary color between cells, which may be useful. Okay. Does everybody have that working all right? The other thing is I mentioned that these cells are not the only thing in the system. There's also a chemical field. There's a growth factor that's being secreted by the cells, diffusing and decaying. And so let's look at that growth factor. If I go here where it says cell field, I can pull down, and there's a menu option which says VEGF, which is a chemical field. Try that. And this in 3D will visualize the isosurfaces of the chemical field. And that may be easier to see in 2D. If I switch back to 2D, I'll see the concentration of my chemical. See if that works for you. How's that going for people? Is it working? Not working? Okay, so we've now run a CompuCell3D. Or maybe gotten stuck for Okay, so there are a lot of things that you could do with virtual tissues. We've talked about understanding basic biology of development, predicting chemical toxicities, potentially designing new drug therapies or making them more personalized. These kinds of modeling that we're going to do in this class are most useful when cells are rearranging, when the cells are doing things as individuals. There are continuum methods to do biomechanics. If I'm going to model the forces on a bone, I don't want to have to model every cell in the bone because there are too many. If I'm going to model chemical transport in the blood, I don't want to model every blood cell. Similarly, I might want to be understanding exactly how the macromolecular machines that copy DNA work. Well, that's a scale that's below what we're doing here. So really, this is a method for simulating cells, what they do, and how they interact with their environment. And over the years, there have been a lot of different things that have been done. These are simulations of a single cell crawling, generating a leading edge and crawling on a substrate. These are old simulations of a tumor growing and interacting with blood vessels. In this case, the tumor is absorbing nutrients from its environment. If you look carefully, you'll see that at the center of the tumor, the cells change color because the cells at the center of the tumor don't have enough oxygen or nutrient, and they die, they start dying. The hypoxic cells send out a signal that causes the blood vessels to proliferate, called nanoangiogenesis. And so this is a simulation in which the blood vessels then are proliferating as they do in a real tumor and supplying additional nutrients to the tumor. That's a beautiful simulation that I wish we had developed a lot more at the time. We published that paper, it got cited a lot, and we never did anything with it. We just let it go. That's an old simulation that I think is really quite beautiful. I tend not to use it. There's a version of it in the demos directory, but running the full simulation on an 800 by 800 by 800, actually 1,000 by 1,000 by 1,000 lattice, it's a bit slow to run it in class. It takes a few hours to run the whole simulation, so it's not so good for educational. We've done simulations of what we call tumor microenvironment, what the selection pressures are that lead to metastasis. That's a simulation one could do. There's a version of that on nanoHUB that you can run. We did a lot of work trying to understand blood vessel development. I mentioned choroidal nevascularization. I love this. It's an old simulation, but I think it's one of the most beautiful ones, just aesthetically, that we ever did. This is a simulation of the retina. Here, the photoreceptors of the retina are made transparent in 3D, so you could see what's going on in the back. You have the photoreceptors here in gray, the pigmented retinal epithelial cells, the black cells that line the eye in green, the membrane that gives structural support to the retina behind the eye in blue-gray, and then the blood vessels behind it. And here, those blood vessels have made a hole in the retina. They've invaded the retina, begun to proliferate, and they're going to then cause the whole structure of the retina to fall apart. This is a disease that I hope nobody experiences. People in the room are probably too young to have this happen. It's called age-related macular degeneration for a reason. It happens more in the elderly. It's actually unfortunately, quite a common cause of blindness in the elderly. One of the things that we discovered doing this simulations and also doing experiments was that the current hypothesis of the day was that this problem was due to excess growth factors. And in fact, the problem is due to failures of adhesion in the retina. That the pigmented retinal epithelial cells that basically have to glue themselves down to their substrate. As you age, there's a layer of lipids, essentially soap, that accumulates on that surface. And of course, it's hard to glue things to soap. And so the cells that line the retina gradually lose their adherence to their support, which is why you're more likely to get a detached retina when you fall as an older person than as a younger person. And that can lead to the invasion of the blood vessels that you see here. And so that actually gave a different approach to trying to treat the disease, because if you don't, many drugs have been successfully developed without understanding how they work. Most drugs that were developed before about the year 2000 were developed without understanding how they work. And there are certainly diseases where we understand how the disease works and we don't have drugs. But in general, it's easier to develop drugs that work if you know how the disease works. And so this was a case where the simulations actually showed that the basic understanding of how the disease works wasn't right. And so for me, that was a big success, quite a nice success. One of the authors here, I guess he's actually fallen off the end of the list here. But one of the authors of that paper was a clinician working on this disease. I mentioned polycystic kidney disease. This is a proliferative disease. It's a little bit like cancer. It's a relatively rare genetic disease. As I say, when you have this disease, your kidneys develop cysts and keep growing until you die. And again, it wasn't really understood very well how that disease worked. And so we were building simulations of essentially the tubule structure of the kidney. One of the problems that you face in homeostasis and maintenance of tissues is that you need cells. There always are some cells that die and you need to replace them. On the other hand, when cells are dividing, you also have to tell them to stop dividing, because if they don't stop dividing, you get tumors or cysts. And so there are a variety of mechanisms, which are typically called things like contact inhibition, which allow the cells to determine the local density of the tissue. If the density is too low, then there's a signal that says divide. If the density gets too high, there's a signal that says stop dividing. That's a nice mechanism because you don't need a blueprint. It's a locally determined thing that maintains tissue density. And angiogenesis, the thing that we saw going haywire earlier, is also a local mechanism that maintains blood supply. You don't need a blueprint. There's a rule that says if I don't have enough oxygen, I send a signal to say, give me blood vessels. That'll work anywhere in the body. The problem is if you're a tumor and you don't have enough oxygen, it works just as well as in normal tissue. Because there's no mechanism that says, don't give me blood vessels if I'm a tumor and do when I'm normal tissue. In the same way here, there are signals that tell the cells in the kidney, if there's a gap, divide and fill in the hole. But sometimes in the people with this particular disease, that signaling is defective, and some of the cells keep dividing when they shouldn't. And that's what you're seeing here is most of the cells in that tube are sitting there. They have the potential to divide, but they say the local density of my tube is correct, and they don't divide. One cell has had the set point for division change, and the result of that is that the cell divides when it shouldn't. Of course, its offspring will also have the set point change, and so they will keep dividing. Now, it winds up that in engineering, we have a principle, and especially in computing, we have a principle of what we call orthogonality. You try to have mechanisms be independent of each other. In this room, if I change the temperature on the thermostat on the wall, I don't want it to change the illumination in the room. I want those to be separate things. I have one controller for the temperature and one controller for the light. In software, you try to design software so that the different modules don't interact with each other in unexpected ways. When you drive, when you put your foot on the gas, you don't want the car to turn. When you turn the steering wheel, you don't want the car to accelerate. That's the fundamental principle of orthogonality. It makes things easier to explain. It makes them easier to understand and control. Biology is evolved. It is not designed. And because of that, there is no reason that biological mechanisms have to be separated in a clear way. They're not designed, and therefore they don't have to be intelligible by us in a simple way. And mechanisms don't have to be separated. And one of the things that we find when we look biologically is that the molecules that have help cells stick to each other also send signals. There's no real reason that that has to be the case. You could have some molecules do the sticking and other molecules sense whether you're in contact or not. But in fact, molecules like cadherins, your CAD8, cadherin 8, are CAD is a cell adhesion molecule. Cadherins are calcium-dependent adhesion molecules. There are many of them. You'll see lots of molecular species with numbers after them because there are lots of different versions of similar molecules. So cadherin 8 is a cell adhesion molecule. It also relays the local cell density. And so this simulation was asking the question, we did with Bob Bacalhau at the med school experiments. You have this great experimental system where you could take human kidney cells, grow tubules with them, introduce what's called an ectopic cadherin, that is a cadherin that's not normally present in those cells, with a colored label, in this case in green. So you could see this cell has normal cadherin, this cell has the strange cadherin, because they're different color. And what he found was you got cysts, and all the cells in the cysts were green, and none of the cells in the tubes were green. So the presence of this ectopic cadherin, this incorrect cadherin, was necessary and sufficient to get cysts. The thing is, the cadherins do two things. They change how much cells stick to each other, and for a given amount of contact, they send different signals to the cell. And so these were simulations to ask the question, was the defect that we were seeing from this improper cadherin primarily affecting how much the cells stuck to each other or how much signal they sent when they were stuck? And this top simulation is changing adhesion, the bottom simulation is changing the signaling. In this case, it was a very clear result that the difference was in the adhesion and not in the signaling. And just as a comment, Bob has now jumped into a company to commercialize that and trying to get clinical trials for drugs based on that observation. Paul and I both did a lot of work on what's called in-host viral kinetics, modeling a patch of lung tissue. Here you start out with a single cell that's infected. You could imagine it being infected. These days, it could be flu or COVID, either one. The cell produces more virus that spreads in the tissue, leads to the infection of other cells. It calls in immune cells. Those cells work by killing the infected cells. In this particular simulation, the virus always gets ahead of the immune cells, and so all the cells get infected and die. This has a rather detailed model of the subcellular viral replication built into it, a diffusive model of the spread of the virus and the chemical signals that call the immune cells, the immune cells' movement and how they kill infected cells. And you can then simulate what happens if you use an antiviral like remdesivir or these days Paxlovid, or some of the newer molecules, and ask the question, if you want it to work, how long do you have after you are diagnosed with the disease? Because one of the problems with antivirals is, especially, for example, the disease like influenza, your viral load peaks just about the time you start feeling sick with influenza. You are maximally infectious before you know you have it. When you are infected with influenza, typically you go around feeling just fine for 2 or 3 days, infecting everybody around you. And then you start feeling sick. By the time you feel sick, and that feeling sick is not from the influenza, that's from your body's immune system, particular interleukin 2 and interleukin 6, which peak and create the fever, headache, and everything else that you associate with flu. That's why people often will say that the vaccination gave them the flu. What they're experiencing is the immune response to the flu. And what you're experiencing then is that you have the same interleukin 2 and interleukin 6 spike, which is what should happen when you have the vaccine, because that's what the vaccine is supposed to do. And so actually the virus itself, unless you're really dying of the infection and you have late stage pneumonia, you're not aware of what the virus is doing to you. Almost all the damage to your lungs is your immune cells coming and killing your own cells to try to get ahead of the virus. And so these were simulations where we said we're going to give different doses of an antiviral at different times and ask the question, since it's a stochastic bubble, green means that essentially all of the replicas of the simulation, we were able to stop the infection before it killed all the cells. Red means effectively all the time, all the cells died. In the middle, sometimes you were successful and sometimes not. That was a very simplified model. TJ took a beautiful model that was developed by Bart Erbentrout and Erica Mohan in Pittsburgh, simulating many cell types because the immune system is pretty complicated, and many chemical species. That ODE model had been extensively validated against detailed experimental time series for how the immune system worked in rodents. And we were able then to translate that into a spatial model of what happens in your lungs when you have a flu infection. So here again, you're seeing that initial infection, the virus will spread here. Type 1 interferons are the first stage of response to infection. Those happen very early in the first few hours after infection. Later on, there are additional chemokines that pull in immune cells, both the innate immune cells like natural killer cells, and then later on the adaptive immune cells like CD4 and CD8 plus T cell. IL-10 comes in at the end and turns off the immune response. And so this was a quite detailed simulation of that, which is pretty realistic in terms, not the geometry, because the lungs, of course, the physical architecture of the lungs is tubes and sacs. And here we've sort of unrolled things to be flat, but the molecular species and the cell types are pretty realistic. We've worked a lot over the years in liver toxicity. This is an area that Maria Bondasson is particularly interested in. Here is a simulation of what happens in your liver when you take Tylenol. Tylenol is carried in the in the blood. It's transferred from the blood to your hepatocytes, to your liver cells. Tylenol itself isn't very toxic. Unfortunately, it's metabolized into something that is quite toxic, a molecule called NAPQI, N-A-P-Q-I, in your liver. Not in every cell in your liver, but in some cells in your liver. If NAPQI binds to a molecule called glutathione, people may have heard of antioxidants, sort of a vague way at some point in life, that are a good thing in quotation marks. So if you have the main antioxidant molecule in your cells is a molecule called glutathione. If that level of glutathione is depleted too much, the cell will die. And so Tylenol is metabolized into a molecule that binds irreversibly to glutathione, depleting glutathione. And so if you get too much Tylenol metabolized into NAPQI, your glutathione levels in your liver drop and the liver cells die. Most of the time, your liver actually is extremely good at regenerating itself. And so probably the first time anybody takes Tylenol, something like 10 to 15% of the cells in their liver die. And they grow back and you never notice it. But if you take too much Tylenol, or if you've been drinking the night before, or you didn't eat anything the night before, your sensitivity to glutathione depletion gets worse. And so the classic person who dies of Tylenol poisoning is either an elderly alcoholic or a fraternity student who drinks a lot, doesn't eat anything, wakes up in the morning with a headache, and then takes extra Tylenol. And every 5 or 10 years, they discover how toxic the stuff is and decide they're thinking about banning it, and then they don't. And there's some good medical reasons why it's useful in other contexts, but it's something that we work with, ironically, with originally with the Environmental Protection Agency, because they were thinking of trying to ban it as a pollutant, and then with the National Institutes of Health, trying to understand the mechanisms of Tylenol toxicity. And there were some people here at IU who worked a lot on it. So those are just a couple of examples of the kinds of models that you could build with this. All of those are CompuCell models, very diverse. Some very simple, some very complex, some with a lot of molecular detail, some with levels of modeling of the whole body, some drug discovery, some basic biology. So that was, and there, I've skipped a lot of slides of different examples. We could go over more. And so I wanted to do now, and we're a little bit behind in terms of the class, so maybe we'll do this as a homework problem, is I would like people having seen these. Some people may come to this class saying, I know what I want to model, which is great. That's fine. Some people may come to this class saying, I don't care what I model. I just want to work with somebody and you tell me what to do. That's fine too. But what I'm hoping is that looking at these examples and maybe doing a little bit of digging in models that are published using CompuCell, PhysiCell, Morpheus, or other systems, there might be something that appeals to you as a problem that you might want to understand. And so I want people to think just for a few minutes about what's a possible system, multicellular system you might like to try to model. If you were going to model it, what kinds of questions might you want to ask? And perhaps most importantly here, why would this kind of modeling be useful? In other words, if you can get away with a model that is simpler, that doesn't have all this spatial detail, go for it. If you need to model the mechanics of bone at a high level with those cells, then you should do that. So the question is, you want to find something where the fact that you have organization as cells and the cells are doing things as individuals is important. And if you have ideas about that, that's great. If you don't have any ideas about that, we'll work with you to come up with something. But it's worth thinking about looking at biological phenomena and asking the question, what are things where cellular organization is critical that are interesting? And so, so because of time, I'm not going to ask you to do that in detail now. Normally, I'd give us some time to do that in class. But I'm going to ask you as a homework assignment to start thinking about some possible questions. What would be a good problem? And if you had a problem, how would you build it? We have these components. We have cells as objects that do things. We have signals that can control the cell. We have chemicals that can be sent between cells. We can have forces that are generated by cells. Cells can migrate. What are the things that your cells would do? Think about this as sort of SimCity for cells. You have objects called cells. You can put them down. They run around. They do things. What are they going to do? How do they signal each other? How do they control themselves? At one point in our lives, we thought maybe we'd try to use SimCity or a version of that as actually a framework for building this kind of model, but it did not. Actually, NVIDIA has this incredible framework for doing video game agent-based modeling in video games and for movies, for Hollywood movies. And we thought at some points about trying to maybe bootstrap on that NVIDIA framework to do this kind of agent-based modeling. So that's something to think about. And as I say, I don't think we're going to probably right now go over that, but as a homework problem, that's a heads up that will be in that homework number 1. So what I do want to do before we break, because we don't have that much time left, is actually write and run a little CompuCell model. We ran one before, but I want to actually run it. So we didn't use the editor. So far, we've just used the player. And so go back to CompuCell, whether it's nanoHUB or your local version. And we're going to turn on, that is a problem, not having enough outlets in this room. At some point in the old days, they had extra extension cords here. I will check to see if I could get extra extension cords for the next class. There are some in back there. I see there used to be a bunch of those, but I probably got snarfed by other classes, I guess. There are outlets there. Okay. So if you are in CompuCell Player, you can go to the File menu and hit Start Quit. Or there's a what is the funny term they use? Discumorph for an icon that shows something that no longer makes any sense, like the floppy disk icon that we have for saving things. So there's a picture of a pen and paper that takes you to the editor. If you're on your local machine, you can also search in your Finder for Twit and launch it directly. So I want everybody to launch Twit. Oops. Thank you. Okay. I want to get our Does anybody need help who's online? I've lost our chat window, so I was trying to find that. Anybody online having problems? Let's see. Did I lose? Was somebody logged into the Zoom? Did I lose the Zoom? It's okay. I just didn't. It's as long as people have it, as long as we're connected, that's fine. Oh, there we go. Yeah. Okay. Thank you. There we go. Okay, got it. I just wanted to get our chat window back. Okay. When I switch screen share from share desktop to just having the PowerPoint, I lose it. Okay, so is everybody able to launch Twit? So you click start Twit, and what you should get is something that that opens a new window. In your desktop, you will have a separate application. On nanoHUB, it will show up as a tab in the X11. And one thing that you may find with nanoHUB is that occasionally nanoHUB will get stuck. There's an X11 bug, which is an X11 bug, where it doesn't redraw the screen properly. And if that happens, if you drag the X11 bounding box and move it a little bit, it'll force a redraw. Nobody said my screen doesn't make sense, so I guess nobody's run into that. But if that happens, you could just toggle the corner. All right. So we are going to start a simulation. And so I want everybody to go to Twit. So everybody should have the model editor up and running. And to begin with, it should be blank. So it should look like this. So I want to make sure everybody's got that. How are people doing online? Got it? I see it. Can other people give me a thumbs up, either virtually or in person? Okay, great. Mohammed, great. Good. Great. Jonathan, great. How about people in the room here? People are Twit running? Great. Okay, good. So session quota exceeded. Did you try to launch a new instance? Okay, so you don't need to launch it from the start. So nanoHUB is a free service. It allows you to run up to 4 processors at once per job and up to 3 jobs at once. So if you go to your back to your dashboard in nanoHUB, it will give you a list of active jobs, and you're only allowed to have 3. So you have to kill the extra ones. Or you go to one of the ones that exists and you can use that. Is that, do you want me to show you how to do that? Or is that something that works? Does that explanation make sense? Sorry? Well, logging in again won't necessarily do it because the jobs will continue to exist in the background. So when you go, the place where you launched CompuCell in the first place? Yeah, I removed them. Okay, great. Then you should do it. But you can have multiple simulations inside a single instance of CompuCell. You don't have to relaunch CompuCell. Yeah, it's not trivial, and the fact that there's slight differences between the desktop and the nanoHUB version make it. They're not very different, but they're just different enough that I have to. People will have slightly different experiences. The other difference is that Windows and Macs have different windows. The windowing systems are different. And so some of the layout and things like that are different on Macs and Windows. And there's not anything we can do about that. That's intrinsic to the way Macs and Windows handle Windows. Okay. So everybody have Twitter running. Anybody not have Twitter? If you have Twitter running, go up to where it says CC3D project, which is the third pull down menu from the top of the top. Pull it down and select new CC3D project. Would people rather have a split screen, or does that make things too small for the moment? Has everybody got that working all right? Okay. Okay. When you say new project, it should pop up a dialog box that says CC3D simulation wizard. Did that work? Okay. So you have to give it the simulation a name. You can type new simulation or something like that. A couple of pointers. nanoHUB is a Linux box. Don't begin a file name with a number or a special character, and don't put any spaces or special characters in the name. Linux hates spaces in names of files, and it really hates special characters in names of files. Underscore is okay, but don't try an exclamation point or a question mark or a space. Over the summer, somebody kept saying, wizard doesn't work, wizard doesn't work, wizard doesn't work. Well, they were creating a file with a 3 underscore something. They were putting a number at the beginning of the file there. Windows is okay with that, but Linux, no. So, okay, so type the file name. It could be new simulation, that would be fine. And then we'll select Python and XML, which is actually going to tell it how to create the simulation. Everybody got that? And then hit next. And that's just going over it again. If you are on nanoHUB, you may want to explicitly remind it that it needs to put the file into your user directory. So you may want to hit browse and make sure your simulation directory is home/nanoHUB/your name. Because if you try to write to the root directory on nanoHUB, you don't have right to it. Everybody got that? I guess I should really be saying, does anybody not have that? That probably be more effectively. Okay. So we'll hit next. And so now it's going to help you walk through creating a simulation. And the first window is called General Simulation Properties. Everybody seeing this window? It asks you how big the simulation should be because it's a lattice simulation. Is it 2D, 3D? Does it have periodic boundary conditions? What's the initial configuration of cells? We're going to just accept all the defaults for today, so just hit next. The next window specifies the types of cells. So we're going to have to explicitly list the kinds of cells that we are going to have in the simulation. And in this simulation, we are going to create 2 kinds of cells. So what I need you to do is go into this dialog box called cell type. Type the name of the cell, could be cell 1, my cell, something like that. Again, no spaces, no special characters, something simple. And then hit the add button. And we're going to do things that a little bit, we're not going to have 1 cell type, we're going to have 2 cell types. So then type a different name. Don't use the same name. It could be cell 1 and cell 2. And again, hit add. So when you're done, you should see something like this. It could say medium, cell 1, cell 2, or whatever names you picked for your cells. Now give people a second. Does anybody need more time on that, or did that work? Logan, did that work? Got it? Anybody here? Everybody got it? Again, I'd rather go slower and make sure everybody's following, and then we can okay. And then hit next. The next menu that you're going to get will say chemical field. And for our first simulation, we're not going to use any chemical field, so just hit next. We're going to skip that. And the next box is going to be, what are the basic properties of the cells that we're going to use? We've said that everything is based on cells and what they do. And you could add a lot more properties besides the ones that we have here, but we're going to start out with the basic one. We're going to say that the cells stick together. So we're going to click this box that says contact. We're going to say that the cells grow. So we're going to click this box that says growth. We're going to say that the cells divide. So we're going to click this box that says mitosis. And we have to say, it seems obvious that the cells have a volume, but in this world, nothing exists unless we assert it. So we have to click the box that says volume local flex. And volume local flex is the name it does because individual cells can be different in their volumes. So we have to say that not all cells have the same volume. They're going to grow. Okay. So you're going to have 4 boxes ticked: contact, growth, mitosis, volume local flex, and center of mass here is going to be ticked by default. Sorry, say that again.
Yeah, I have one type of surface and one type of volume.
Okay, so when you say that cells are going to grow, you have to allow the cells to change their volume individually. So it grades it hides the option of
Oh, yeah,
global volume. Yeah. So this is what you see before you tick that box. When you hit growth, that will disappear. Good question. Anybody else? Everybody got it? So now hit next. And you'll get a message saying configuration complete, finish, hit finish. And now you'll be back in your editor. You should see a line here that says CC through whatever you call your simulation. Double click on that, and it should open it up, and you'll see the simulation code that was generated. Did that work for people? Did it not work for anybody? Is anybody stuck? Got it? No? Everybody? Okay. So, to save the simulation, we will use the obsolete icon 2 floppy disks, or case floppy disk. This one here, which is save all. The first one just saves whatever window is open in front. The second one is for renaming, is a save as. The third one is save all. You can also pull down the CC3D project menu and hit save CC3D project. So hit save. You'll find that there are a lot of different ways to do the same thing. That makes it convenient because different people like doing things in different ways, but it also means if I'm showing you, I won't necessarily show every possible way of doing the same thing. Okay, if I want to run the simulation, I can right-click on the simulation tab here, and that will give me an option that will say open in player. I can also pull down in CC3D project, there's an open in player option. If I am in desktop, it will open player directly. If I'm in nanoHUB, I will have to click the tab at the bottom of the X11 that says player. So I'll give everybody a minute to do that. I could do that. I could do that in okay. Let me do this with you. Put it. This is the simulation. Okay, so here we have the simulation. I hit open in player. I now switch to my player window here. And I should see the simulation around. Did that work for people? Okay. This simulation runs pretty fast. So by the time you get anywhere, it probably has already grown all the way through. But I could hit, in this case, I told you don't hit stop. But here I'd hit stop, hit step to get it started. This is my initial configuration. And now I hit step, you'll see the cells grow. If I zoom in, I can see the cells grow, they divide, when they get to a doubling volume. And the reason we have two cell types is that when the cells divide, if they are type 1, they become type 2. If they are type 2, they become type 1. So they swap back and forth between the cell types. Okay. How about our online people? Did that work for you? Yes, good. All right. So you've written the CC3D simulation. Yes, go ahead. Mm-hmm. So in other words, it just showed one cell type here, and you created two cell types. Okay, so that probably is, as I say, there definitely are bugs in CompuCell, and getting the online version of CompuCell updated is somewhat cumbersome. So that may well be a bug that it doesn't display the cell colors properly. So send me an email and I will see if we can get a newer version of CompuCell uploaded to Nano. Thank you. No. Okay, so we have exactly 4 minutes to go. So I want to talk for just a second and actually show you what CompuCell code looks like in 4 minutes. Don't worry, we will not. I am not assuming that you know how CompuCell code works after 4 minutes. CompuCell code will typically consist of 2 parts. XML CC3D ML code that specifies the static properties of the cells and the simulation. How big the simulation is, how many processes you're using, what the cell types are, and so on. And then dynamic things are going to be scripted in Python. And we could look at that in more detail. I'm going to skip through this because we don't have that much time. If we look at the XML, the first two lines specify number of processors, debug output frequency, dimensions of the cell ladder, here 256 by 256. How long the simulation runs, 100,000 steps. There's a sort of a mysterious property called temperature, which is correlated with a particular methodology used. It's the fluctuation amplitude of cell membranes in the simulation. And something called neighbor order, which is also a technical issue about the methodology used to do the cell movement. We're going to typically ignore that, although it doesn't. We'll talk about it when we need it. Then we have a list of our cell types. Here we have something called medium, which is the empty space around the cell. And whatever you call your two cells here, cell 1 and cell 2. And then the cells stick to each other. So we have the contact energy between medium and medium, between cell 1 and cell 2, between cell 2 and cell 2, and so on. Here, by default, they're all the same. But in fact, we could adjust those if we want to have things like different coherence. And then at the end, we have an initializer. CompuCell is not that great in terms of the number of initializers we have. Here, uniform initializer draws a square or a rectangle and fills it with cells. And we can say how big the cells are, how many cell types we want, whether we want them to be touching each other or not, and so on. The next thing we have is the Python code. And the Python code, for people who are used to Python, is a class. Yes. I'm sorry, say that again? Yes, yes. Yes, you can, well, there's something called PIF, which is an initialization file that specifies the configuration. So you could create a simulation initializer that specifies the configuration. Or you could go into your Python layer, read a file in Python, do it by hand. Or you could use Python to draw some complicated pattern if you wanted. Okay. Again, if you don't, if the Python looks unfamiliar, don't worry, we're not going to do a lot with Python at the moment. You'll see that all of the simulations have three components, two of which we'll use a lot. There's a start function, which is called once at the beginning of the simulation, that we use to initialize things, and then there'll be a step function that's iterated every time you step the simulation. In the start function, we have to do things like define how big our cells are going to be and how compressible they are. We don't get anything for free. So here we're saying our initial target volume for our cells is 25, and their compressibility factor is 2. And we'll talk next week about what those mean. And you'll see something that's very Pythonic, which is for-cell in self.cell list. CompuCell will keep a list of all the cells in the simulation, and if I want to do something to every cell, the syntax is for-cell in self.cell list. So that Python you'll have to learn and use the colon to define the ending of Python. For people who are not Pythonic, indentation is used instead of parentheses for defining blocks of code. The good news is that Twinit will do all of this for you so you don't have to remember it. Twinit has what are called code snippets. So if I say, I want to do something to all cells, I say, Twinit, give me the code for doing that. It'll paste it in. Doesn't mean you don't have to know a little bit of Python, but okay. Then in the step function, we're going to say our cells grow. For cell in self.cell list, cell.target volume plus equals 1. Plus equals adds, instead of saying cell.volume target volume equals cell.target volume plus 1, plus equals adds 1 to them. And you could play with that. When cells grow, we have to have a divide. This is a little bit of Python, which is maybe not so obvious. You have to create a place to keep a list of the things you're going to divide. So a square bracket in Python says you're going to create a list. You're going to create a list called cells to divide. You look at the cells. If the cell volume is bigger than 50, we add the cell to the list. Then we run through the list of cells to divide, and we say divide them. And we're going to divide them with random orientation. If we were doing bacteria, we divide them along the long axis. Sometimes we could divide them along the short axis. We could divide them along cleavage planes that we define. We have a lot of options. So we divide them along a random direction. And then when we create a new cell, we have to say what happens to it. And we say that the child cell, self.clone parent child, says the child cell inherits the properties of the parent. Except we divided the volume by 2. We're going to allocate half of the volume of the parent to the child and half to the parent. And then the one little trick we have is we say if the parent was a type 1, the child is a type 2. And if the parent was a type 2, we make the child a type 1. So the colors will alternate. And that's the simulation we ran. And that is, we're over by 5 minutes, so I don't want to keep you more. But that is your first engagement with CompuCell. I know it was a little bit of a rush at the end, but you could go home and play with it. You could change the doubling volume of the cells. You could change the growth rates.